Hello, everyone. I'm Kristin Mars. I'm a sequencing specialist here with PacBio, and I'd like to welcome you to this webinar series. So I just wanted to show my face very quickly and say hi to everyone, and we'll go ahead and get started. So I would like to introduce our panelists today, and it's my pleasure to welcome them. We really appreciate them lending their expertise to the discussion. So first off, we have Melissa Laird-Smith from the Econ Institute of Data Science and Genomic Technology in New York City. Next, we have Olga Veneer-Peterson from SciLife Lab in Sweden. And finally, we have Michael Hardigan from the University of California in Davis. We have a lot of really great material to cover today, and the presentation portion of the webinar will be followed by a panel discussion and a questions and answer session. You're welcome to submit your questions at any point during the webinar by typing them in the area provided on your attendee dashboard. We will also be recording this webinar and making it available for download in the next few days. I also want to point out that we have provided you with 3 useful sequencing handouts that you can download now from your attendee dashboard. So with that, let me get started by giving you an introduction to the PacBio technology and its applications. Please note that I'll be asking 3 polling questions during the course of my presentation, so please do participate in those. And we are actually going to go ahead and get started with a polling question. This just lets us know a bit more about your sequencing background, so I'll give you a couple seconds to answer this question now. All right, so it looks like a good majority of you, over half, currently use sequencing in your research. So this is going to be a really great webinar for you, and we'll go into a little bit more detail about the different sequencing technologies that you can use. So in this presentation, I'm going to give you a brief summary of the evolution of sequencing. We'll discuss how PacBio long-read sequencing works, and then we'll go into more detail about what you can actually do with PacBio long-read sequencing. And then at the end, we will have a panel discussion with our experts. So looking at the evolution of sequencing, I'm sure since most of you said you're familiar with sequencing, that you're very familiar with first generation sequencing, which was Sanger sequencing. So Sanger sequencing is a method of DNA sequencing based on the selective incorporation of chain terminating dideoxynucleotides by DNA's polymerase during in vitro replication. Most notably, this was used during the Human Genome Project. With this, the process is relatively slow and expensive, and the read lengths were quite short. You're generally working with fragments that are between 500 to 1,000 base pairs, but it is an accurate sequencing technology. Next came second generation or next generation sequencing. So this again was a short read sequencing technology. It worked through high throughput from the parallelization of sequencing reactions. It did have generally short reads, so anywhere from 50 to 500 base pairs, but it was able to bring sequencing to a broader population because it allowed for much more fast and affordable sequencing. And then after that came third generation sequencing, or what you may consider long read sequencing. So since this is long read sequencing, we're working with much longer read lengths, on average tens of kilobases of fragments. We're also sequencing native DNA with third generation sequencing, and we're looking at that in real time at a single molecule resolution. And one thing you may have heard is that traditionally long read sequencing has a lower accuracy than NGS. And one of the things I'm going to cover today is how PacBio has been able to overcome this lower accuracy to provide you both highly accurate and long read sequencing data. So next we have our second polling question, and we'd like to get a sense of what type of sequencing you've used in your research. Okay, it looks like the majority of you are very familiar with short read sequencing. So this is going to be a really great webinar to start dipping your toes into long read sequencing and specifically PacBio sequencing and what it can do for your research. So let's dive into that in more detail. So when we talk about PacBio sequencing, we have a couple key advantages that I would like to highlight in the beginning. The first one is the one that you're most likely familiar with, and that is that we are a long read sequencing technology. When I say long reads, on average, our read lengths are greater than tens of kilobases, but we can sequence inserts anywhere from 500 base pairs all the way to much greater than 50 kb. So really any application is suitable here. We also are a highly accurate technology, and that is due to the difference between us and other technologies is that PacBio is free of systematic errors. So any random errors that occur within the sequencing, we're able to wash out easily to get a high consensus accuracy of greater than Q50 or 99.999% accuracy. We're also looking at the data in a single molecule resolution. So each read is going to come from a single DNA or RNA sequence. And with that single molecule, we're going to get you a really high accuracy of greater than Q20 or 99%. So it's going to be really useful for your downstream analyses. And also, compared to other technologies, PacBio is going to have very uniform coverage across any species that you're working with. There's no DNA amplification that's required either before sequencing or on the system itself. And we're going to be able to provide the least amount of GC content and sequencing complexity bias. So if you're working with an individual that has a really high amount of repeats or complexity to it, PacBio is going to be able to span those regions much better than any other technology. And another cool advantage, because we are looking at native DNA in real time, we're able to do simultaneous epigenetic detection, and this comes at no additional cost or sample preparation to you. So let's look a little bit deeper in the workflow of this. So you're going to start off with your sample of interest, whether this is a virus or all the way up to much larger vertebrates. You'll isolate your DNA or RNA as you do with all sequencing technology. And then you'll go into the PacBio library preparation, and we call this a SmartBell library. And as you can see, we are ligating on hairpin adapters to either your DNA or cDNA. And then that has an addition of a primer and polymerase that goes straight onto our sequencing instrument, the Sequel II. And then that single molecule is going to be sequenced in its sequencing vessel, which we call a zero mode waveguide, or ZMW for short. So to zoom into that ZMW just a little bit deeper, you can see that it's a well that is going to contain that one single molecule of either DNA or RNA. And so we're actually diffusing that single molecule into the ZMW, and the polymerase is bound to the bottom. So we're watching that polymerase incorporate phospholinked nucleotides in real time. And then we're able to convert that into base calls and include the kinetics that are associated with that. And so you can see that with these hairpin adapters, that creates a topologically circular molecule that when you look into it, we're able to go around that one single molecule multiple times. So we're going to get multiple passes around the forward and reverse strands, creating a very long polymerase read. And then we're then able to break that polymerase read down into one single, very highly accurate read. And you're going to have uniform coverage throughout that entire read through low complexity regions, and there's not going to be any amplification bias that comes with it. So when you thought about sequencing previously, there were two kind of paradigms that you could think about. So one of them was if you wanted really high accuracy, you had to do short reads. And so those reads are going to be very short, generally much under 1 kb. Or you could do long read sequencing, and with that you were able to get read lengths out past 50 kb, but the accuracy for those was going to be a little bit lower than short read technology. Well, with PacBio and our long insert highly accurate reads, we're able to provide a new paradigm to you where we can provide both really long reads, that are highly accurate. So you don't have to pick between short and accurate and long and less accurate. We now have one data type for you. So how long are HiFi reads? Well, on average, HiFi reads are going to be around 20 kb. So these are still much longer reads than you're used to working with short read technology. This is an example of an HD002 library that was sequenced with our current chemistry. So with a 20 kb read, we're able to span really complex regions, but we're still able to go around that molecule many times to get you a high accuracy. When I say high accuracy, how accurate is that read? So this is a just under 20 kb HiFi read that we had a predicted accuracy of Q33. When we actually dive in and look at this read, we see that there are only 8 errors in a just under 20 kb read. So that comes out to a 99.96% accurate read that's on a 20 kb scale. So this is going to be a really powerful data source for your downstream analyses. And why does accuracy matter? Why should this be important to you? When you think about base level accuracy, that's going to allow you to start to differentiate between real variation and errors. So when looking at a human assembly, one thing I like to point out is this: with PacBio HiFi reads, you're able to go in with a much lower coverage than you need with other technologies. So for a human assembly, we only needed 22-fold coverage of this HiFi data to be able to get us a really high accuracy of Q51. And if you compare it to other technologies, either LongRead or LongRead plus short read, they're not able to come anywhere close to that accuracy as with PacBio. And then looking at the correctness of the reads, looking at RICE, again, we were able to go in with just 20-fold coverage. We're able to get a really high BOOSCO score of 98.7%. This is going to allow you to see more complete and frame shift free genes in your genome, so you can actually trust the data moving forward. So we're gonna come to our last poll question, and we're curious, what is more important to you, long read lengths or high accuracy? All right, that is the answer I would like to see. It looks like both of these things are important to most of you, and I will completely agree with that. Most of us at PacBio believe that you need both long reads and high accuracy for your research. So let's dive into that a bit deeper. So what can you actually do with these highly accurate long reads or HiFi reads? Well, first of all, you can take these HiFi reads and go in to do your genome de novo assembly, and you're going to get the most complete assembly across any of the technologies. You can also take these HiFi reads and do variant detection, and no matter if you're looking at very small SNVs to indels or much larger structural variants, you're able to call all of them with a very high precision and recall. You can take these variants and phase them into haplotypes. And when we look at that from a human space, we were able to detect 5% more variants in the medical exome. And these are specific genes that are relevant to human health and disease. Then in the RNA space, you can sequence full-length transcriptomes with very highly accurate reads with our IsoSeq pipeline, which I'll go into more detail. And also when looking into the metagenome space, the HiFi reads become really important because you need both the read length and accuracy to be able to fully look into your complex population. So one of the things we get asked a lot at PacBio is HiFi sequencing expensive and time consuming? And the answer is simply no. With the Sequel II system and the SMRT Cell 8M, you're able to get a very high throughput sequencing at a very high accuracy in a short amount of time and at an affordable price. We have very efficient workflows for you, starting with our library prep. We have one sequencing kit or one template prep kit that can be used across the board for multiple applications and a one-day library prep. The sequencing is generally done in one day, and our data analysis is very fast. You can actually do most genome assemblies with HiFi data in under a day. And in terms of cost, we've really brought down the price of sequencing with our Sequel II system. Per SMRT Cell, the cost is about $1,300, and that is included with library prep and sequencing reagents. So what can you do with that one SMRT Cell at a $1,300 cost? Well, you can do quite a lot. One of the main applications is you can actually sequence a whole genome in one SMRT Cell for any genomes that are up to 2 gigabases. You can saturate an entire human transcriptome in one SMRT Cell. When looking at targeted sequencing, the sky is basically the limit here. You can multiplex as many samples as you would like into one SMRT Cell. We actually recently had a group that multiplexed up to 36,000 samples into one SMRT Cell. So really there are no limitations when you look at the targeted approach. And with complex populations, HiFi again is a very important data type for this. You can actually multiplex up to 96 16S samples into one SMRT Cell. And if you're looking at profiling communities, you can multiplex up to 3 communities in one SMRT Cell. So we do have a one SMRT Cell menu, and this is one of the documents that we provided for you in this webinar. So I would highly encourage you to download that document and look in a bit more detail at what you can do at this low cost for one SMRT Cell. So I'm going to dive into each of these applications briefly, starting with whole genome sequencing for de novo assembly. So I don't think I need to tell a lot of you, but there are a lot of challenges associated with genome assembly, starting off with just the size and complexity of various genomes. The human genome itself has over 3 billion base pairs. And then we start to look at plants, they can often have much larger genomes. And not only are they larger, but they're very complex. A lot of them have extreme repeat content. Maize and wheat are just 2 examples of this, and wheat has over 80% repeat content. And then each project is going to be a bit unique. Each genome is going to have a range in size, ploidy level, and heterozygosity. So you generally needed a custom strategy for each genome, but PacBio's made a very simple approach for you to take for any genome assembly. So let's talk just a little bit about short read genome assemblies versus long read HiFi genome assemblies. So with short reads, you're not going to be able to expand these really complex regions that are within the genome. So when you go to do your assembly, you're going to have a lot of gaps present in them. So you're not going to have a complete genome, you're going to have what we call a draft genome. And this missing sequence is going to lead to missed genes, and it's also going to very much limit your biological interpretation. Now, compared to doing a genome assembly with our HiFi reads, we're going to be able to give you the most complete comprehensive genome possible. There's going to be no gaps in there, so you're really going to not be missing any data within the genome. We're known to give you the most high quality gold level reference genomes. And with chromosome size context, you no longer really need to worry about scaffolding. So with these HiFi reads, you're able to span those repetitive elements that we briefly discussed. And this can be any type of repetitive element, whether you're interested in transposable elements or flanking direct repeats, terminal inverted repeats. Because these reads are both very long and highly accurate, we have no issue spanning those repeats. And then one of the key benefits is that you're also going to be able to phase those haplotypes. So with short read data, everything's going to be collapsed into a single mixed sequence. But with HiFi PacBio data, each haplotype is going to be assembled separately, so you get the most complete picture of your genome. So we do like to grade ourselves on our genome assemblies, and by grading ourselves, I mean looking at the 3 C's of contiguity, completeness, and correctness. So you can see that HiFi assemblies do really well on all fronts. We're going to give you a very continuous assembly. It's going to be near complete, and the accuracy is going to be very high. You can see this example is at a Q47. So when we look at this in a bit more detail and compare ourselves to the Genome in a Bottle benchmark, so looking at this, we broke our assembly into 100 KB windows and mapped those against the HD002 call sets. And we're only looking at those high-grade confidence calls for accuracy. And if you look at this with an average of Q48, 40% of that genome was perfect. And you're not able to see this with any other technology. So PacBio HiFi data is really going to be able to provide you the most complete, accurate, and continuous genome assembly out there. Now moving on to variant detection. So when we talk about variant detection, there are many types of variants within your genome. They can be very small, anywhere from single nucleotide variants to in the mid-size range with deletions and insertions. And then they can span to very large structural variants within the genome. So when you look at these on a scale, generally you had to break these up into 2 different technologies. So to be able to find those shorter variants, the SNVs, which are 1 base pairs, or the indels, which are less than 50, you needed a really high accuracy to be able to call those. So you had to use short read technology. And then for structural variants, which are greater than 50 base pairs, you need the long reads to be able to span that entire structural variant to properly call that. So you had to use long read sequencing. Well, now with our HiFi reads that are both very long and highly accurate, you only need 1 data type to go in and call all of these variants. So it's a much more approachable application, which is 1 data type needed to give you a very high precision and recall of all variants. And looking at how those HiFi reads map, so this is the STRC gene. You can see that with short read data, you have a lot of unmapped regions, so you're not going to be able to call those variants in that area. And then with ONT, you can see that it does map to that region, but there's a lot of errors in there, so you don't know which variants in there are real. With PacBio HiFi reads, not only can we fully map to that region, but also to phase those variants. So it's going to give you a really clear picture of your genome and be able to call those variants accurately. Now moving on to RNA sequencing. So in RNA sequencing, when you're dealing with short reads, you essentially take your cDNA and you break it up into much smaller pieces and try to parse that back together. But with this, there's going to be a lot of gaps. You're not going to have any reads that span all of the junctions. So it's not going to provide you a very clear picture of your transcriptome. Whereas with PacBio HiFi reads with transcripts, we're going to be able to fully capture all of the transcripts within the system and be able to characterize all of the splicing events that are happening. With PacBio, we call our full-length RNA sequencing iso-seq, and we are sequencing the full-length cDNA. So when I say that, I mean all the way from 5' N to poly A tel. And because we are sequencing the entire transcript, there's no assembly required with RNA sequencing. So it's a very easy approach for this. And with this, you can discover novel genes and isoforms, and you can do this even in very well characterized samples. A lot of people have been using this to improve their genome annotation, and this can be done with or without a reference. And another really great approach is that you can improve the accuracy of your RNA-seq quantification at isoform level resolution. So I already mentioned that you can saturate an entire human transcriptome in 1 SMRT cell. We also have multiplexing options. You can multiplex up to 12 tissues into 1 single SMRT cell. And you can do RNA sequencing in a targeted or whole transcriptome fashion with as little as 30 nanograms of RNA. So to touch base on just 3 examples really quick, we have the Brahman and Angus cattle project. And with this, they were looking at both allele-specific and tissue-specific isoform expression. Recently, a preprint came out from a cannabis group that was looking at tissue-specific transcripts of both THC and CBD synthesis. And they were also able to go in and do the genome annotation of the Y chromosome. And then in the human space, in the cancer transcriptome, we've had groups that have looked at identity fusion genes, and then they're able to use this data and compare it with the genomic findings that they have previously had. So metagenome space, this is another application that really benefits from the HiFi reads. And we actually have 3 options for you, but when you look at this from short read versus HiFi reads and metagenome, with the short reads, you're only going to be getting partial genes, and so your genomes are going to be very incomplete. With HiFi reads, since they're both long and highly accurate, and we're going to have multiple genes per read, we're going to be able to give you the best reference quality assemblies from that sequencing data. And as I mentioned, we have 3 approaches when you're interested in looking at complex populations. The first one is looking at full-length 16S sequencing. And you can do this in 2 different ways. We have a PacBio one-step protocol that allows you to multiplex up to 96 samples together. Or you can use Shoreline Biomes kits, which is an all-in-one kit that does extraction, amplification, and analysis. And if you're asking why is it important to look at full-length 16S as opposed to just subregions, a paper recently came out of the Weinstock lab highlighting this importance. So if you look at this, most people often go in with short read technology and just look at subregions of the 16S gene. And with that, there can be a lot of bias, and you're not always able to classify all of the taxa within that subregion. So a lot of the sequences get thrown out. When you look at just the V4 region, which is a very common region that's sequenced with 16S, in the chart on the left-hand side, you can see that the red is the higher percent of unclassified sequences. The V4 region, there's a very high percentage of unclassified regions. Whereas if you look at the V1 to V9 representation, which is the full-length 16S, you have very little to no unclassified sequences within there. So you're really getting a complete picture. And to quote the paper, sequencing the entire 16S gene provides real and significant advantages over sequencing commonly targeted variable regions. The next approach, if you're interested in looking at complex populations, is to do shotgun profiling. And the real value here is that with PacBio reads, they're going to be on average of 10 kb with our HiFi data approach. And with a 10 kb read that's at a Q20 and above accuracy, you're actually going to be able to get a predicted 8 genes per read itself. So when you think about this comparing to short read technology, you're generally taking short reads and you're having to assemble those together for metagenome analysis. And our HiFi reads alone are going to be longer and more accurate than the genome assemblies that you get from short read data. And you're actually able to take just the HiFi reads alone and do gene discovery directly on them. And because these reads are so accurate, you can plug them into existing NGS tools and pipelines. So it's a really easy, efficient way for you to do gene discovery. And lastly, if you're interested in doing metagenome assembly to create new references, you can easily do this with our HiFi data type, and you're going to get really highly accurate complete genomes with as little as 15 to 20-fold coverage. And another cool advantage of this is that you can actually leverage the epigenetic data, and you can cluster contigs and plasmids from the same strain together. So it's a very powerful data type for complex populations. And so in conclusion, I would just like to reiterate that long-read sequencing is needed to gain comprehensive views of whatever you're interested in looking at, whether it's a genome or a transcriptome. But it does matter to have both long and accurate reads. Not all long reads are going to give you the same benefits in your research. And these HiFi reads can be used across the board for different applications and sample types. And this is going to be able to better enable your biology and discovery. So we're really excited about this HiFi data type, and we've already seen a lot of great research coming out with this. So with that, we'll now go ahead and move over to the panel discussion. All right, I'm just trying to get my webcam on. Here we go. All right, so I'd like to welcome again our panelists. And we'll go ahead and get started. So we had a lot of people submit questions when you registered for the webinar, which thank you ahead of time. And so we're going to go ahead and start off with a question of what factors do you use to determine if a short read or long read approach is the best for a specific project? And Olga, I'll go ahead and pass this along to you for you to take on.
All right, thank you, Kristin. So good afternoon or the good time of the day to everybody. Right, so answering this question is actually the part of my job. So I would put it this way, everything depends on the scientific question of the project that you are working with. So first of all, we start, for instance, with RNA-seq. If you are interested in splice isoforms, so there is actually no good way to assess the all-comprehensive wellness of all the isoforms in the cell unless you're really using long reads. However, if you are looking for differential expression studies, perhaps PacBio technology is not the perfect application for you yet. However, I hope that you guys are working with that, so it's something that comes along the line. Another thing is that if you are interested in certain genes of interest, now you're not biased to 300 or 500 base pair amplicons anymore. So theoretically, if you would like to sequence genes that are up to 13 kb long, here definitely PacBio would be a really good option. Very low-hanging fruit is, of course, de novo sequencing of genomes. But again, for some of the projects, you do not need to have those long reads. But if you're asking some certain types of evolutionary questions, or if you're looking for non-Mendelian traits, there definitely you would need to have the long reads, and they have a great advantage as compared to the short reads. If you're looking for structural variants that are longer than 50 base pairs, long reads is definitely the way to go. If you want to phase your variants, again, you would need your long reads. And yeah, so just every project is unique. So I would say that's a scientific question that decides.
Awesome, thank you, Olga. Was there anyone else from the panel that wanted to chime into this question?
Sure. Yeah, in our lab we're breeding focused. We use long reads basically to try to get the best possible genome assembly quality we can, and then layer short reads on top of that for marker discovery and then developing markers and deploying them within the breeding program. So for us, it's pretty straightforward in that regard, but we're also starting to look into other ways that especially the new HiFi technology can benefit us. Big interest for us sort of in the pan-genome direction is sort of trying to look at the pan-genome of resistance genes in particular, because that's a very clear target for us, where that technology can really elucidate things where you have regions with a lot of genes that are evolving quickly. There's a lot of tandem duplication, things like that. So there's a lot of interest in both areas for us.
Great, thank you, Michael. All right, I will move on to the next question. So this question is sample preparation, DNA extraction, and library prep for long-read sequencing complicated? And are there any tricks to get the best results? And so I'm actually going to pass this one again to Olga, because I know she does a lot of work on this, and will be able to hopefully provide some tricks for you guys.
All right, no pressure. So PacBio technology relies on the native DNA because there is no amplification step. So for that, you would optimally, to achieve those beautiful long HiFi reads, you need to have high molecular weight DNA. And currently, there are tons of different kits on the market that can help you with that. For instance, Circulomics Nanobind can vouch for them. Not for every tissue type, not for every organism, but for very many tissues and organisms, that's a great kit. MagaTract from QIAGEN, beautiful kit, works perfectly on vertebrates and some other species. There is Mesheray Nagel's nuclear spin columns. They are excellent for plants and for fungi. And also recently, I know that, I don't know if Kristin, you're going to show that, there is an excellent resource that you can find through the PacBio's homepage that will guide you through the extraction procedures, specifically for PacBio sequencing. Also, I would like to give a shout out to protocols.io. They have an excellent group for high molecular weight DNA extraction for any type of organism. And finally, if you're on Slack, there is a group called ATTUTA, ATUTA, all things up to assembly. There are lots of tips and tricks of extracting really great quality DNA. for PacBio applications. But the length is not the only thing. Your DNA should also be chemically pure. And some of the kits, they will remove part of these contaminants that can interfere with the sequencing later on, but not all of them. So in some of the cases, you have to go to the old school molecular biology, the way we did it in the 80s and 90s, when you extract really good high molecular weight DNA. But speaking again, tricks, if you're working with plants or anything with high sugars, extracting nuclei is an excellent way to go, and then to extract DNA from nuclei directly. Try to extract from as fresh sample as possible, both for RNA and DNA. I'm working as a part of the Vertebrate Genome Project. That's a part of the Earth Biogenome Consortium, and we're actually working on the paper with set of instructions for people who are going out to the field to collect samples and bring them to the lab for reference genome sequencing. So we really hope that this paper will be out sometime in spring. So we're working on that. There will be more tips and tricks. But that's from the top of my mind. Sample fresh, as fresh as you can, as long molecules as you can, and as pure as you can. And there is lots of information online nowadays that you can find.
I'd like to add something to that. I think not only the methods that Olga mentioned, but how you assess the quality is critically important. So when you're thinking about how to QC your samples to assess whether they're good for PacBio sequencing or whether you're going to submit them to a core like any of us run, really focusing on using methods that specifically quantify and qualify double-stranded DNA is critical. So we use the Qubit HiSense kit, double-stranded DNA kit, is our gold standard. Please, please, please try to move away from the nanodrop. The nanodrop instruments quantify any nucleotide in a solution, so that would include any fragmented, nicked, degraded material, primers, the NTPs from a PCR. So really choosing the right QC method is really frustrating both for users as well as us operating the system. If you think you're submitting sufficient amounts of DNA, but really you're submitting far, far less because the QC methods are different between the groups. So don't hesitate when you're talking to service providers or talking to PacBio to really nail down the auxiliary support systems that will help you get and qualify your best samples.
Exactly. Thank you, Melissa and Olga. And Olga, we're very excited for that paper that you referenced. And I do actually have a slide at the end of this to list the website that you mentioned from PacBio that has a long list of DNA extraction methods that have worked for various users, and it's broken up by species. So I will shout that out when we get to that page, but it's a very useful tool for you. All right, I think we can move on to the next question. So this question is, what depth is necessary to overcome sequencing errors, and how do you assess accuracy and validate long-read sequencing data? So Michael, do you want to go ahead and start us off with this question?
Sure. Yeah, so we've been in an interesting position because we've had several rounds of sequencing, even across multiple technologies, going back to the RS2, to sort of see how things pan out. And what we've seen is with the assemblers we've been working with, we work primarily with the Falcon assembler. Below 70x coverage of raw data, we start to see significant drop off in quality or genome completeness. So that seems to be a pretty important point for us. That could, I'm sure that could fluctuate depending on your species. We work with an octoploid, and usually if we start off with, say, 90x subread coverage, the post error correction, the corrected p-read coverage we're usually looking at is 25 to 30x. So we see a significant drop off there. We're still able to get good assemblies with 25 to 30x corrected read coverage. But yeah, I think for us that 70 to 80x range seems to be a threshold we need to get above to get good results. As far as looking at the accuracy and validating the long-read sequencing data, upstream of assembly, we've been looking at a couple of things to try to look at the accuracy of the long reads. We have short read data, and we've been looking at trying to do some k-mer analysis to look for things that might be artifactual. But we actually haven't gone too far in that direction yet, to the point where I can say this is how much it's affecting the results of our assembly. Downstream, we're really focusing on the things that you brought up during your talk, as far as looking at coverage, completeness, and correctness. We've seen significant improvements going from short read data to PacBio data as far as assembly completeness. It adds another layer of difficulty where you start separating out haplotypes, and addressing that heterozygosity and getting back to the target haploid assembly size has presented challenges, but we found ways. I think we're going to talk about that next, but we found pretty good ways to get around that as well. So
great, thank you, Michael.
I'd love to jump in and add some context also from some of the work we do. I mean, Michael's working with an incredibly complex genome. So when we work with diploid genomes, with human genomes, we try to generate about 25x per allele coverage for haploid level resolution. So we aim for 50x coverage of diploid genomes. But most of the work we do is targeted sequencing. And so we use a lot of the HiFi intramolecular accuracy and consensus accuracy modes. So again, as Kristin mentioned, when you have an insert where the polymerase goes around multiple times and then you can collapse those reads to wash out error, the accuracy you can achieve with CCS or the HiFi algorithm is incredibly high on the Sequel II. In fact, it's higher than some of the RTs and polymerases we use to generate the amplicons to begin with. So we've actually gotten into situations where we're characterizing a lot of PCR error, which is a blessing and a curse to some extent. But a little bit, it's very interesting to see, a little bit scary when I think about all the PCR work I've done in the past. And I think that validating that data, you validate in any way you validate any sequencing data. An orthogonal method, if you really want to qualify that variant that you're interested in there, is the way to go. So as Michael mentioned, a short read validation, any other orthogonal validation is how we would really confirm, if needed, that this was here. I think also, you are doing single molecule sequencing, so these are phased single molecules of DNA. So seeing it in multiple different molecules, sampled multiple times within an extensive run, is also a nice way to validate in an internal fashion.
Yeah, if I can build on that as well, something that we're really excited about with the HiFi data is basically with this sort of consensus technology, being able to get past that error correction step. So as you can imagine, when you have four subgenomes all within your genome that are maybe 2 to 5% divergent sequence-wise, and you have subreads that have maybe 10 or even 15% sequencing errors, it's a bit of a black box when you're talking about doing all-on-all error correction. You don't know quite what the potential is for an area where those subgenomes are very similar to actually error correct what are truly different homologous sequences in those different subgenomes. Based on the completeness of the assembly output, we think that actually it does a better job than I had hoped for originally. So that's really good to see. But with these HiFi reads, the data we've seen from you guys, where we align it back to our reference and see over 99% identity, that's beautiful, because it takes that initial step out where it is a bit of a black box, where you're not exactly sure what's happening in the error correction stage involving separate but very similar subgenomic sequences, to saying I can just move almost directly to the assembly step and just say, well, I don't need to loosen that threshold for aligning these things for error correction. I can set a very strict threshold that would exclude subgenomic sequences in the first place. So from that standpoint, I think the HiFi data for polyploid species genome assembly has just a huge potential for reducing errors that could occur in that initial correction stage. So.
Absolutely. So we did have a question come in, so I was hoping one of you could expand on. When you were talking about the coverage with your recommendations, were you talking about for HiFi or traditional long-read sequencing in terms of the actual coverage that you need to overcome the sequencing errors?
I'll start with that. I mean, that's a really great question, first of all, and it is a critically important factor to address because the HiFi coverage requirements are still being developed by PacBio recommendations and still being sort of empirically tested in the labs. I think the coverage that Michael and I were both talking about were a CLR or the long insert subread coverages. The recommendations for HiFi read coverage for doing variant detection are sort of, 15 to 20x coverage for a human genome at this rate, and we're seeing very good concordance with that with the data we've run so far.
And
yeah, I agree. I think if we can get decent assemblies with even down to 20x coverage corrected, 20x coverage in the corrected reads based on the subreads. So I think the HiFi reads, which already have low error rates, again, I think this still needs to be determined. But if the error rates are as low as a corrected P read based on the existing technology, I think you could expect to hopefully see similar results with a similar coverage. So I think we could probably get a similar genome quality with 20 to 30x coverage of HiFi as opposed to, we like to start out with like 90x coverage of the uncorrected subreads. So I think that drastically would reduce the requirements there.
Absolutely. All right, well, thank you for clearing that up. I think we're ready to move on to the next question. So this question is, how do you address heterozygosity in complex plant genome projects? And can you use long reads in highly repetitive DNA like maize or to sequence homopolymers? So I'm going to give this one to Michael since he works with a very complex plant to go ahead and take the lead on this question.
Sure. Yeah. So for octoploid strawberry, when we just put in the data and don't try to do anything particularly to parse out haplotype phases upstream of the assembly process, usually our assembled genome size is about 160% of the haploid genome size. So we were, not only is our species octoploid, but the subgenomes themselves are very heterozygous. So we know we're separating those haplotypes and getting two contigs for a lot of haplotypes in the genome. So we've actually moved towards trying to bin our data sets into phased bins to do parallel assemblies upstream, and we found a lot of luck there. I think there's been a few things that have studies that have come out where they've used trio binning using Illumina sequencing of the parents to try to parse out reads into phase 1, phase 2 bins and then separately assemble. We're working with things, right? So we're interested in that. Maybe out of a lack of foresight, when we first decided what we were going to try to assemble, we picked one individual that has incomplete parent information and another individual that's a wild native California beach strawberry, which if it's a wild individual has no parent information, obviously. So we've actually been able to go back to more of an old school method where we do skim sequencing of a mapping population, and we were able to do recombination breakpoint prediction and map and phase about 4 million variant sites across the genomes. And based on those, we're able to actually extract the flanking sequences around those variants and phase them, and then use those phased. We use 2 KB sequences flanking mapped variants, and those phased flanking sequences we can actually use in the same way you would use Illumina for trio binning and parse things out into 2 pools. And when we do that, we've been really happy to see our assembly output is exactly the haploid genome size, the predicted genome size. based on flow cytometry. So I think, especially with these polyploids, you need to try to find some way to do that upstream, because we've actually had pretty bad luck getting the, I mentioned we use Falcon to assemble, unzipping and purging haplotigs into, two even-sized sort of contig pools has not worked well for us. And I suspect that that has something to do with the fact that it's an octoploid and not a diploid. So, sorry, I've probably gone over time. I'll let somebody else jump in if you'd like to.
No, that was great. Thank you, Michael. Did anyone else want to jump in on this question, or I can move on? All right. So our next question is, can long-read sequencing detect genetic lesions such as FMR1 CGG repeats or HTT CAG repeats? So I'm going to give this one to Melissa. I know she's done some work on both of these repeat regions.
Yeah, so the short answer is yes, and actually I've done work on this with Olga. So this is a long-standing collaboration. So these are incredibly difficult regions to resolve in the genome because of the repetitive content. They're not easily amplified, so you can't really do them with the PCR strategy. Current methods in the clinic still use Southern blot, which for obvious reasons is not ideal, in addition to the fact that you don't get nucleotide level resolution. So we've been able to do particularly the FMR1 CGG repeat using the PacBio NoAmp method. They revised their NoAmp method to work with SQL and released a new protocol not long ago. It's pretty robust, and they actually have primers recommended for that repeat as part of that protocol. With HTT, we've actually done it with an amplification method. So I think this is sort of a nice way to segue into sort of targeted methods overall. Often for your gene of interest, you may not want to sequence the entire 3 gig human genome to look at your one stretch of genomic content that you're interested in. I work in a lot of polymorphic immune loci with my own research. And so there's a lot of different target enrichment methods you can use. You can use amplification. You can use some of these NoAmp methods that PacBio has developed. You can use oligo-based pull downs, somewhat akin to whole exome sequencing with short reads, where your oligos are complementary to your region of interest. And in this case, for almost all of those, you would use the HiFi reads, so using intramolecular consensus to resolve the complexity of those regions, to reduce the error profile, which works quite well. So in the case of the HTT repeat, we actually did this. And in this case, the region that we were looking at, we were trying to phase the actual CAG repeat with a SNP upstream. And in DNA space, those are separated by about 200 kb. So even that is too long for a PacBio read. So in this case, we targeted it in the RNA space. So we generated total cDNA and then amplified this region using a gene-specific primer set. And it made about a 10 kb amplicon. And in that case, we're able to sequence it and generate high-quality data that then could phase the SNP 10 kb upstream from the repeat and associate how long the repeat was with the carriage of the SNP. And so I think there's a lot of different ways you can target sequences. The random error mode that Kristin described earlier that PacBio Sequencing provides does not stumble on a lot of these types of repeat contexts within the genome, which means you can cleanly resolve them. The last one I want to mention as sort of a use case is we've also sequenced the C9orf72 repeat. So this is a G4C2 hexanucleotide repeat associated with ALS. And we've actually done whole genome sequencing on an ALS patient and found an over 900 repeat locus there. So that's G4C2 900 times. And we were able to sequence through it with a single PacBio read for the very first time that we know of. And again, getting a little bit towards the low complexity or homopolymer question that was posed earlier, the fact that we can get through these types of regions speaks very highly for the stability of the sequencing across any genomic complex in the genome. And Olga definitely has comments.
Yes, I have one comment, completely with you on that. One thing is the beauty of long-read sequencing is that not just you can read through those low complexity regions, homopolymers, you can also decipher the epigenetic signature in them. So in case if you want to look at the base modifications, how they bind, how the complexity is, how they're regulated, that's actually amazing thing. So that's for the first time we can actually start to observe how the epigenome is working in those things. So.
And just to comment, that is for the native DNA, so the NoAmp method, the whole genome sequencing. As you amplify, you erase those epigenetic marks. but it's known in the case of FMR or suspected that epigenetic modification is critical for the pathogenesis of disease. So I think that's a very good comment. It's an added benefit you get from every sequencing run of native DNA on the platform.
Absolutely. Thank you for that. And so this unfortunately is going to be our last question. We're almost at the hour mark. So I did have one last slide that I wanted to provide you with just a bit more information. So all of the applications I touched based on today, you can find on our website as well. And the website that Olga mentioned, extractdnaforpacbio.com, is a really great resource if you're just starting off in your project and you're trying to find the best extraction method for your species. Please do take a look at that. And also, if you have any really good extraction methods that have worked well for you, please let us know. We'd like to add it to the list. We also refresh our publication library with every new publication that comes out, so you can always find the latest and greatest there. If you are interested in doing a project, we have a lot of certified service providers out there. So these are sites that have our instruments that have been pre-approved by us. They all do very excellent work. So you can find them on our website, and they'll be able to walk you through the entire process from library preparation to sequencing. A lot of them do the downstream analysis as well. So it's a really great option for you. Also, if you're interested in purchasing a system, we recommend that everyone purchases one or two to have on hand for all of your HiFi needs. And lastly, we have a lot of events going on at all times, so you can stay up to date on our latest events, whether it be a conference. We have a really exciting roadshow coming up called HiFi Live. This is actually going to be in 9 locations. And I know that Melissa is speaking at quite a few of these, so please find the one that's closest to you and go ahead and register. There's going to be a lot of really great talks there. And additionally, Olga has a long-read sequencing seminar coming up on 10/12 that you can register for at UPSLA. I'm sure there's going to be a lot of great information provided there as well. And so with that, I would like to thank our panelists, Melissa, Olga, and Michael, for joining us today and sharing their expertise. We really do appreciate it. And I did want to briefly mention 2 things. So this webinar has been recorded and will be available for download very shortly. So keep an eye out for that email with the recording. And also following the webinar, we're going to have a very brief questionnaire. So please do take the time. It's only 2 minutes to fill this out. And this is going to provide a lot of information for us at PacBio to help us develop these webinars. So again, thank you very much for joining us today. We really appreciate your time, and we can't wait to see what exciting research you do with PacBio. So thank you again.
Thank you.
Thank you.
Bye-bye.
Bye, everyone.